Welcome to the deep dive. Today we're plunging into something absolutely fundamental, a process that really underpins modern medicine. How the drugs we rely on are conceived, created, and eventually make their way to us.
It's a journey that is, well, probably far more intricate and layered than most people imagine. It's not just about scientific discovery, right?
Right.
It's this complex tapestry woven from history, cutting-edge tech, enormous financial risk, and pretty dense regulatory and policy frameworks.
And for this deep dive, our source material is straight from the bedrock of the field, the first chapter of Goodman and Gilman's The Pharmacology. This isn't just any textbook, it's like the text.
Yeah.
The one that defines the terrain.
Yeah, absolutely. Think of this chapter as the essential primer. Before you dive into the specifics of how drugs interact with the body, G&G first lays out how we even arrive at these things.
You mean like the invention, development, regulation?
Exactly, and the whole ecosystem of industry and government that surrounds them. It sets the stage.
Our mission today, then, with this incredible source as our guide, is to unpack that foundational knowledge. We're going deep into the evolution of drug discovery, the nuts and bolts of the modern process, the gauntlet of clinical trials, regulation, and the fascinating, sometimes contentious, policy side. If you follow this field, this is your shortcut to understanding that whole pipeline from the G&G perspective.
It's a challenging area, definitely, but also, fascinating. A real intersection of pure science, engineering, business strategy, and public health.
Okay, let's unpack this. Let's start right at the beginning. How did Goodman and Gilman frame the field in their very first edition, way back in 1941? How did they define pharmacology?
Well, that 1941 definition, it cast a really wide net. They described pharmacology as covering the knowledge of the source, physical and chemical properties, compounding physiological actions, absorption, fate and excretion, and therapeutic uses of drugs. Pretty comprehensive.
And their definition of a drug itself was super broad back then, wasn't it?
Oh, yeah, incredibly broad. Any chemical agent that affects living protoplasm, by that definition, lots of things could be drugs.
Things we wouldn't normally call drugs today.
Exactly. But while that's the academic starting point, the practical reality and what the rest of the book really focuses on, it quickly shifts to the legal definition.
Which narrows it down quite a bit.
Totally. A drug, in the sense of something you can actually get prescribed or buy, is a chemical or biological agent that's been rigorously evaluated and approved by a national regulatory body like the FDA here in the US or the EMA in Europe. That's the universe G&G mostly explores.
Got it. So this first chapter, it sets the stage. It covers the pipeline before they get into the later chapters on mechanisms, how drugs actually work. This is the how we get the drugs chapter.
So precisely. It gives you that essential context for everything else that follows in the book. It's foundational.
And the history of how we've gone about finding or creating these agents is quite a story, an evolution.
It really is. The earliest phase, as G&G lays it out, is deeply rooted in, ancient human experience with the natural world. Plants, mostly.
Makes sense. Plants are just full of chemicals, and people must have noticed their effects.
Exactly. The source gives those classic examples, Coffee berries and caffeine for stimulation. Atropa belladonna extract for dilating pupils that gave us atropine. Mahuang for ephedrine. Curare for paralysis and hunting. And poppy juice, obviously, for opium and morphine.
Pain relief, diarrhea treatment.
Right. But also recognizing the addicting properties early on, like with nicotine, cocaine, ethanol.
Yep.
They mention those two in this historical context.
And the key thing about this early era,
the key insight is that these discoveries were based purely on the observed biological effect. You chew this bark, your fever goes down, you eat that berry, you feel energetic. There was zero understanding of how it worked.
No concept of a molecular target or anything like that.
None. It was purely empirical observation, trial and error, sometimes dangerous trial and error, discovering toxicity or addiction the hard way.
And this search for natural products, it didn't just stop in ancient times, right? Antibiotics.
Oh, absolutely. Penicillin, cephalosporins derived from fungi. They're prime examples from the modern era. Basically, chemical warfare agents made by microbes that we learned to harness.
But this natural product era started to shift with the rise of synthetic organic chemistry.
Yeah, and interestingly, it owes a lot to the dye industry in the late 19th century, making dyes.
Dyes. How did that connect?
Well, dyes showed this remarkable ability to selectively stain certain biological tissues, but not others. That selectivity was a huge clue.
Ah, okay. a hint that chemicals could be specific.
Exactly. It was for Paul Ehrlich. Anyway, he studied these dyes and came up with the idea that cells must have specific chemical receptors that selectively bind these compounds. He then thought, well, maybe we can create magic bullets.
Magic bullets.
Chemicals that would selectively target pathogens, like bacteria, without harming our own cells, based on this receptor idea.
And did it work?
It led to a massive breakthrough. His work culminated in arsphenamine, also known as salvarsan, in 1907. It was the first really effective treatment for syphilis, a game changer.
Wow. So it proved the concept.
It did. And then later, Gerhard Domack discovered that Prontosil, which was actually a dye, was effective against streptococcal infections. That launched the whole sulfonamide era, the sulfa drugs, and really kicked off antimicrobial chemotherapy as we know it.
So you see this pivot happening from just observing nature to deliberately making compounds and testing them, guided by these new ideas about receptors.
Yes. And by the mid-20th century, this crucial collaboration between academic pharmacology, synthetic chemistry, and clinical medicine really solidified. The experimental approaches started evolving rapidly alongside our understanding of biology.
Like moving beyond testing only in whole animals.
Right. Early discoveries like, say, indomethacin and NSAID were found by testing compounds in animal models of inflammation. But as we understood disease mechanisms better, down to the cellular and molecular level, we could develop in vitro assays.
Testing in glass.
Exactly. You could say, purify an enzyme thought to be involved in a disease, put it in a test tube, and then screen lots of synthetic compounds to see if any inhibited its activity.
That must have been a huge leap forward.
Monumental. It meant researchers could test far more compounds, much faster and more efficiently. And it started to give medicinal chemists clues about what kinds of chemical structures were showing promise against a specific molecular target.
Okay, here's where it gets really interesting. Because alongside those biochemical assays, structural biology was taking off too.
You mean figuring out the 3D shapes of proteins?
Exactly, using techniques like X-ray crystallography, NMR spectroscopy, starting to get these high-resolution pictures of proteins, often with small molecules actually bound to them, especially from the 1980s onwards.
And that must have been transformative for designing drugs.
Absolutely. For decades, we knew drugs bound to proteins, but the atomic details, total black box. Drug design was often, very empirical, hit or miss.
But with the 3D structure.
You could actually see the binding site, the pocket, and you could start to rationally design molecules that would fit snugly, interact in just the right way, aiming for high affinity, high specificity. That's structure-based drug design, SBDD.
And this lined up perfectly with computers getting way more powerful.
Which was essential. I mean, just getting and refining those structures needed serious computation. Visualizing them interactively was new. And crucially, it enabled computer-aided drug discovery, KED.
Right. Using computers to model how drugs and targets might interact, manage all the data, guide the design process.
Things like molecular simulations, predicting how molecules move and bind.
Yeah, simulations, sophisticated databases for chemical and biological info, and eventually high-throughput screening, HTS using robotics to automatically test massive chemical libraries against those in vitro assays we talked about.
Okay, and the latest layer on top of all that is AI, artificial intelligence.
Right, applying things like deep neural networks to analyze these huge data sets, predict binding, even help design molecules automatically, or predict protein structures themselves.
So this modern process, it's really a fusion. Structural biology, massive computing power, automation, AI, all built on that foundation of chemistry and biological assays.
Precisely. Which leads back to that point G&G makes. Are we still discovering drugs, or are we really inventing them now?
Given that whole process you just described, it feels much more like invention.
It really does. The term drug discovery totally fits the historical context, finding active stuff in plants or microbes. But like the chapter says, most new drugs today are synthesized compounds. They're meticulously designed, iterated on, optimized through that whole complex interplay of design and experimentation. They are arguably invented chemical entities.
Okay, let's dive into that modern process for small molecule drugs then. Where does it typically kick off?
Well, most projects start with basic research. Work that points to a specific macromolecule, usually a protein, as playing a key role in a disease.
Okay.
The underlying idea is that if you can modulate this protein's activity somehow, you could treat the condition. So that protein becomes your candidate drug target.
And the drugs would either boost its activity or reduce it.
Exactly. A lot of small molecules are inhibitors or antagonists. They reduce the target's function. Think statins inhibiting HMG-CoA reductase for cholesterol.
Or antibiotics hitting bacterial enzymes.
Right, like beta-lactams inhibiting bacterial transpeptidases. Others are activators or agonists, often for signaling proteins, like albuterol activating beta-2 adrenergic receptors in asthma.
So how do researchers actually find these initial target candidates? Where do they come from?
It's multiple routes, really, often overlapping. Sometimes an existing drug or even a natural product reviews the target, like penicillin pointed towards bacterial cell wall enzymes. Other times it comes from deeply understanding a disease pathway. You map out the steps, see a critical enzyme, and target that, like the cholesterol synthesis pathway pointing right at HMG-CoA reductase.
Or sometimes from studying whole families of proteins involved in disease.
Oh yeah, that's a major area. Kinases, for instance. These are enzymes that add phosphate groups to other proteins, sort of like switches. They're often hyperactive in cancer.
Right.
So understanding kinase function and finding specific kinases driving cancer growth led to inhibitors like imetinib, which targets that mutated BCR-ABL kinase in CML.
And these omics technologies must play a huge role now.
Yeah.
Genomics, proteomics.
Absolutely. They've been game changers for target ID. Genome-wide association studies, GWAS, compare genetic variations across huge populations, people with and without a disease.
Okay.
And they can find correlations between specific gene variations and disease risk, which strongly suggests the protein product of that gene might be a good target.
And transcriptomics, looking at mRNA levels, proteomics for protein levels.
Yeah, comparing diseased versus normal tissues using those methods also points you towards proteins that are up or down regulated, suggesting they're involved.
And then you combine all that omics data with digging through databases, the scientific literature, patents.
Crucial data mining, yeah. Pulling it all together. But, not all target identification starts with knowing the protein. Phenotypic screening is still a thing.
You mean testing compounds just to see if they have the desired biological effect without knowing the specific target up front?
Exactly. Maybe using automated microscopy to screen chemical libraries for compounds that, say, kill a pathogen in culture or make cancer cells behave normally or whatever the desired phenotype is.
And then you figure out how it works.
Right. If you get a hit, the next challenge is target deconvolution, figuring out which protein or pathway that hit molecule is actually interacting with.
Like that Plasmodium falciparum example in the book.
Yeah, good one. Growing malaria parasites under pressure from a drug, then using omics to see how they develop resistance. Those resistance mechanisms often point straight to the drug's target protein.
Okay, so you've got a candidate target protein. What's the really critical next step before you pour millions into designing drugs against it?
Target validation. G&G really emphasizes this. You need stronger evidence that hitting this target will actually impact the disease in a meaningful way. And just as importantly, that messing with this target won't cause unacceptable side effects because of its normal function in the body. That's on-target toxicity.
Basically de-risking the project.
Precisely. And there's no single magic bullet test for validation. You use a range of methods. Maybe chemical probes, highly selective small molecules that mimic the drug to study the biological effects of hitting the target. Chemical genetics is big here.
Or you can use genetic approaches.
Yep. Things like using siRNA to silence the gene, basically mimicking inhibiting the protein's production, or using genetically modified mice knockouts where the genes are deleted, or transgenics where it's altered to study the gene's function in a whole organism context.
The Box 1-1 example on leptin is, wow, a really powerful lesson on why validation is so crucial.
It truly is. Leptin looked like the perfect target for obesity based on mouse studies, right? These mutant mice were obese, lacked leptin. Give them leptin, they get thin.
Seemed obvious.
But it turned out most obese humans actually had high leptin levels. They were leptin insensitive. So hitting the target or giving more leptin didn't fix the core problem. The biology was way more complex, redundancy, compensation. That's a hard-learned lesson driving this need for really thorough validation. You don't want to chase a target that won't work in humans.
And while you're validating, you also have to think about whether the target is even druggable, right?
Yeah, that's a key question, especially for small molecules. Can it actually bind a small molecule tightly enough? You check public databases like 9DB or PubChem, see if similar proteins bind ligands. You look at 3D structures in the protein databank. Does it have a nice pocket or cleft where a small molecule could fit?
These pockets seem really important for small molecule drugs.
They are. They're common in enzymes that naturally bind small substrates or receptors for small hormones or neurotransmitters. But proteins that interact with other large proteins, they often have big, flat surfaces.
Harder for a small molecule to grab onto effectively.
Much harder. You don't get enough specific interaction points. That makes them less druggable by small molecules, and it's a major reason why protein therapeutics, like antibodies, have become so important for those kinds of targets.
And I guess there's this continuous effort to map out all the potentially druggable targets in the human proteome and even figure out ways to tackle those previously considered undruggable.
Absolutely. The ultimate validation, of course, is actually developing a successful drug that works via that target. That's your first-in-class drug. Like Merck's lovastatin, the first statin, it validated HMG-CoA reductase as a target.
But not every drug project aims to be first in class. You mentioned Me Too drugs.
Right. A lot of drug development focuses on already validated targets, targets where a successful drug already exists. These Me Too drugs might seem less innovative on the surface.
But they can still be valuable.
Definitely. The goal is often to improve on the first drug. Maybe better potency, a cleaner side effect profile, easier dosing, trying to become best in class, like atorvastatin, Lipitor, which became a massive drug even though other statins existed before it.
So Me Too isn't necessarily bad. It's about optimizing against a proven concept.
Exactly. Though, as we'll probably discuss later, it does raise questions about the balance between true innovation and just incremental improvements in the industry.
Okay, let's touch briefly on targets that aren't just single proteins.
Right. Polypharmacology. That's where a drug interacts with multiple targets, sometimes intentionally, sometimes not. It's pretty common when targeting proteins within a family where the binding sites might be quite similar, like adrenergic antagonists or many protein kinase inhibitors.
When might hitting multiple targets actually be a good thing?
Well, if the targets are maybe in the same pathway, hitting both might give you greater efficacy or overcome some biological redundancy. Or maybe hitting two distinct targets is beneficial for a complex disease. But designing a single small molecule to do this predictably and selectively, that's really hard. Systems pharmacology tries to understand these network effects.
But hitting multiple targets can also cause big problems.
Oh, yeah. Unintended polypharmacology hitting things you don't want to hit, these anti-targets, is a major source of side effects and toxicity. The classic example G&G mentions is the HERG potassium channel in the heart.
The dreaded HERG channel.
Exactly. Inhibiting it can lead to potentially fatal arrhythmias, so compounds get screened very heavily early on to make sure they don't hit HERG. It's a major liability flag.
It's also worth remembering, like G&G points out, that some small molecules don't even bind to proteins at all.
True. Good point. Examples include platinum-based anti-cancer drugs like carboplatin. They work by directly damaging cancer cell DNA. Or aminoglycoside antibiotics. They bind to bacterial ribosomal RNA, not protein, to shut down protein synthesis. And antiviral nucleoside analogs get incorporated into viral DNA or RNA, messing up replication.
And then there's that really unusual drug, sugammadex.
Yeah, fascinating mechanism. It's basically a modified sugar molecule, a cyclodextrin. Its job isn't to treat a disease, but to reverse neuromuscular blockade during surgery.
Wow.
It literally finds and encapsulates the paralytic drug, rocuronium, in the bloodstream, just traps it. This lowers the free concentration of rocuronium very quickly, reversing the paralysis, a completely different kind of drug action.
Okay, but for the majority of small molecules that do target proteins, what are the absolute must-haves for binding?
High affinity, non-negotiable. You need the drug to bind really tightly to its target.
Why is that so critical?
So that a low dose can be effective. If it binds tightly, you don't need a high concentration floating around, which minimizes the chance of it binding to other off-target proteins and causing side effects. Affinity is often measured by the dissociation constant, KD.
Lower KD means tighter binding.
Right. You're typically aiming for a KD around 1 nanomolar, or even better, sub-nanomolar, if possible.
And affinity isn't enough. Specificity is just as important.
Absolutely. You want that tight binding only to your target protein, not to thousands of other proteins in the body. Avoiding off-target binding is key for safety. And sometimes the kinetics matter too, how fast the drug binds, and, maybe more importantly, how long it stays bound, its residence time. That can influence how long the effect lasts.
What actually holds the drug and protein together? What are the forces?
It's primarily non-covalent interactions, things like hydrogen bonds, attractive electrostatic forces between opposite charges, the hydrophobic effect where non-polar parts of the drug and protein like to snuggle together to avoid water, and also short-range dispersion forces, sometimes called van der Waals forces.
It sounds like a delicate balance. These forces have to overcome other things.
They do. They have to overcome the natural tendency towards disorder, entropy, and they have to overcome the energy cost of stripping water molecules away from both the drug and the protein surface as they come together. So yeah, achieving high affinity is a complex, energetic balancing act.
And you mentioned earlier that binding usually happens in pockets, not on flat surfaces. Why?
Because pockets allow for lots of these short-range interactions all around the drug molecule. A flat surface just doesn't offer enough points of contact for a small molecule to bind tightly and specifically. That's why pockets are common in enzymes and small molecule receptors, and why those protein interaction surfaces, often flatter, are tougher targets for small molecules.
The chapter also talks about allosteric binding. What's that?
Ah, yeah, that's really important. Allosteric means the drug binds to a site on the protein that is different from the main functional site, like the active site of an enzyme.
Okay, so not competing directly with the natural substrate or ligand.
Exactly. But binding at this distant allosteric site causes a change in the protein's overall shape, its conformation. And that change in shape affects the function of the main site. It could either inhibit it or sometimes even activate it.
Can you give an example?
Sure. The non-nucleoside reverse transcriptase inhibitors for HIV, like efavirenz or nevirapine, they bind to a pocket away from the active site, but lock the enzyme in an inactive shape. Another example is ligands that bind to allosteric sites on GABA A receptors in the brain.
And this allosteric approach can be useful for getting selectivity.
Hugely useful, especially within a protein family where the main active sites might be very similar and hard to target selectively. The allosteric sites are often less conserved. They might be unique to one family member. So targeting an allosteric site can be a clever way to hit just the protein you want and not its close relatives.
And then finally, there are some drugs that actually form covalent bonds with their targets.
A smaller number, but yes. They form a permanent, irreversible chemical bond, usually reacting with a specific amino acid side chain, like a cysteine, in the binding pocket.
What's the advantage of that?
Well, because it's irreversible, you might be able to use smaller doses or dose less frequently, since the drug doesn't just dissociate and get cleared. The effect lasts as long as the protein does.
But isn't there a risk? Forming covalent bonds sounds potentially dangerous.
There absolutely is a risk. The big danger is that the reactive part of the drug might react with other proteins it bumps into, leading to off-target toxicity or immune reactions. So selectivity is absolutely paramount here.
How do you get selectivity with a reactive molecule?
Usually, it relies on precise non-covalent interactions first. The drug has to bind specifically in the right pocket, positioning its reactive group perfectly next to the target residue before the covalent bond forms. That initial specific binding prevents it from reacting randomly elsewhere. The example of sotatercept is really elegant here. It targets a specific mutant form of the KRAS protein, G12C, found in some cancers. It works because that mutation introduces a cysteine residue, the C, at position 12. Sotatercept forms a covalent bond specifically with that cysteine, but it doesn't react with normal KRAS, which doesn't have a cysteine there. Very targeted.
Okay, so we've covered identifying targets, understanding binding. Now, the actual lab work to find a molecule that does all this, what's the ultimate goal of these experimental approaches?
The goal is to arrive at what's called a clinical candidate. That's a molecule that looks promising enough to actually test in humans.
And what makes it promising enough?
It needs to hit all those marks we've discussed. High affinity, high specificity for the target, producing the desired biological effect in preclinical models. But that's not all. It also needs to have favorable ADME or DMPK properties.
ADME?
Yeah, absorption, distribution, metabolism, excretion, or drug metabolism and pharmacokinetics. Basically, how the body handles the drug. Can it get absorbed into the bloodstream? Does it go where it needs to go? How quickly is it broken down or eliminated? Does it turn into toxic metabolites?
Okay, so good binding isn't enough. It needs the right PK profile, too.
Absolutely. And on top of that, it needs to show an acceptable safety profile in preclinical tests, meaning lab assays and animal studies. And from a practical standpoint, you need to be able to actually manufacture it reliably, purify it, and formulate it into a stable form, like a pill or an injection.
That sounds like a massive checklist, a really tall order.
It is. It requires a huge amount of initial in vitro testing. checking things like, can it get into cells? How susceptible is it to being broken down by liver enzymes? Is it toxic to cells? Does it cause mutations?
But G&G makes the point that in vitro tests alone aren't enough, right?
Definitely not. They're essential for early filtering, but they don't fully predict what will happen in a complex living organism.
So animal studies are still mandatory before human trials.
Yep. Typically required in at least 2 species, usually a rodent like a rat or mouse, and a non-rodent like a dog or monkey. You're looking closely for any signs of toxicity and also gathering critical pharmacokinetic data in vivo, how the drug behaves in a living system.
And all this is happening while chemists are figuring out how to make the stuff efficiently and formulate it.
Right. Manufacturing scale-up, cost, purity, stability, formulation, those are huge practical hurdles too.
Okay, the chapter highlights something interesting. While we've gotten much better at finding molecules that bind tightly ligand discovery, designing in good safety and pharmacokinetics from the start is still really tough.
Yeah, that remains a major challenge. Because safety and TK involve incredibly complex interactions. Your drug might interact with thousands of different proteins and processes throughout the body in ways that are very hard to predict just from its interaction with the intended target. It's a much bigger biological puzzle.
Medicinal chemistry seems like it's right at the heart of this whole optimization process.
Oh, it's absolutely core. Medicinal chemists are the architects and builders of these molecules. They're part of a big team, obviously, but they're the ones who actually synthesize the compounds, purify them, figure out if an idea is even feasible to make.
They guide the design based on how hard it is to synthesize.
Yeah, synthetic accessibility is huge. How many steps? How expensive are the starting materials? Does it create tricky mixtures of isomers? And they have this deep chemical intuition about how changing a little part of the molecule, adding this group, removing that one, might affect all those properties we talked about.
Like how it interacts with the target, how it gets metabolized, whether it can cross the blood-brain barrier.
Exactly. They use strategies like making biososters, swapping one chemical group for another with similar size and electronic properties to try and improve metabolism or avoid toxicity, for example.
And they use rules of thumb, like the rule of 5.
Yeah, Lipinski's Rule of 5 and others provide guidelines about the sorts of properties like size, polarity, that tend to correlate with good oral absorption. But experienced chemists know these are just guidelines, and there are many important successful drugs that break these rules. They're heuristics, not laws.
Okay, so one major engine for finding those initial starting points, especially if you don't have a target structure, is high-throughput screening, HTS.
Right. HTS is the industrial scale approach. Think automation, robotics, tiny wells on plates, testing huge chemical libraries, sometimes hundreds of thousands, even millions of different compounds against your biological assay, usually looking for an optical signal.
Huge numbers.
Yeah.
But the success rate isn't great.
The hit rate is typically very low, maybe 0.1% or even less.
Ah.
And a lot of those initial hits turn out to be false positives for various technical reasons. So you need really careful data analysis and confirmation assays.
And even the real hits, they're not usually drugs right out of the box.
Almost never. HTS hits are just starting points. They often bind weakly. They might hit lots of other things. They probably have terrible PK properties. They need a lot of work.
Optimization. Which brings us to SAR.
Structure-activity relationship studies.
Yeah.
Once you have a hit, you start making and testing closely related compounds, analogs. Maybe you buy similar ones, analog by catalog, or, more often, chemists synthesize specific variations. You change one little piece at a time and see how it affects potency, selectivity, solubility, metabolism, everything.
Trying to figure out what parts of the molecule are important.
Exactly. Figure 1.1 in G&G gives a nice visual example, showing how tiny changes to a molecule drastically altered its activity against different enzymes. That SAR data guides the chemists on what to make next. It's an iterative cycle. Design, synthesize, test, analyze, repeat. Often hundreds of compounds are made.
And the best compounds emerging from this process become lead compounds.
Right. Those leads then go into lead optimization, which is an even more intensive phase focused on fine-tuning all those properties, potency, selectivity, ADME, safety, trying to get everything just right to produce that clinical candidate. And hopefully some backup candidates too, in case the main one fails later.
What if HTS doesn't give you good starting points, or you want to explore chemical space differently?
That's where fragment-based drug discovery, FBDD, has become really popular. It tackles the problem that the number of possible drug-sized molecules is just astronomically huge, like 10 to the power of 60, maybe more. HTS only samples a tiny fraction of that chemical space.
So FBDD looks at smaller pieces.
Exactly. It screens libraries of much smaller molecules, fragments. These fragments usually bind very weakly to the target, often too weakly to show up in a standard HTS assay. But you can detect their weak binding using sensitive biophysical methods like X-ray crystallography or NMR spectroscopy.
And the advantages?
You can see exactly where these small fragments bind on the protein surface. Even weak binding tells you that a particular chemical shape or feature is compatible with that spot. Then you have a couple of strategies. You can find two different fragments binding near each other and chemically link them together into a larger, higher affinity molecule. Or you can take one promising fragment and start growing it, adding bits chemically to improve its interactions and affinity.
So it's a more targeted way to build up a lead.
Yeah, it avoids that combinatorial explosion of randomly testing huge molecules. It focuses the chemistry effort based on validated interactions, even if they're initially weak. G&G mentions vemurafenib, a melanoma drug, as a notable FBDD success story.
And beyond HTS and FBDD, there are other cutting-edge experimental technologies emerging.
Always. Driven by the constant need for more efficiency, lower costs, better predictions. One big area is DNA-encoded libraries, or DELs.
DELs? What are those?
Imagine screening not millions, but billions or even trillions of compounds simultaneously in a single test tube. That's DELs. Each unique chemical compound in the library is physically attached to a unique short DNA sequence that acts like a barcode.
Okay. So how do you screen them?
You mix this huge library with your target protein, which is usually immobilized on a bead or surface. You wash away everything that doesn't bind. Then you collect the molecules that did stick to the target, amplify their DNA barcodes using PCR, and sequence the DNA. The sequences tell you exactly which chemical structures were the binders.
Wow. That's incredibly high throughput.
Massively so. It's a powerful way to explore enormous chemical diversity and find starting points.
And what about efforts to make the in vitro testing more predictive of what happens in humans?
Yeah, that's the clinical trials in a dish concept. The idea is to move beyond simple flat layers of cells and culture, which often don't behave like tissues in the body.
So using more complex models.
Right. Using human pluripotent stem cells, you can now generate specific types of human cells, liver cells, heart cells, nerve cells, etc. And then you can assemble these cells into more realistic three-dimensional structures called organoids, or even use techniques like 3D bioprinting to create small pieces of artificial tissue.
And the hope is these models better mimic human physiology.
That's the goal, to get better predictions of a drug's activity, its metabolism, its potential toxicity before you ever get to human trials, reducing animal testing, and hopefully improving the success rate in the clinic.
Okay, so those are the experimental approaches. Hand in hand with those are the computational methods, SCADs.
Computer-aided drug discovery. Yeah, information technology has completely revolutionized this field. Just the ability to store, manage, and share these massive data sets is huge. Plus, the software and the sheer speed of computation keeps increasing exponentially.
Chemical informatics plays a big role. Managing chemical data.
Definitely. Building databases, searching for similar molecules, relating structure to properties, and we now have these incredible public databases, Shemble, BindingDB, PubChem for chemical structures and bioactivity data, the Protein Data Bank for 3D macromolecular structures, GenBank for sequences, UniProt for protein information, even pathway databases like Reactome and KGG. All this interconnected data fuels the computational models.
And the ultimate dream for computational methods is?
High accuracy prediction. Being able to computationally predict, just from the structures, exactly how tightly a potential drug will bind to its target, and maybe even predict its selectivity against all other human proteins, its ADME properties, its toxicity, all before you even synthesize it.
Are we there yet?
Not quite, but the predictive value is definitely increasing. The underlying physics is incredibly complex. Ideally, you'd solve Schrödinger's equation for the whole system, but that's computationally impossible for drug-sized molecules interacting with proteins. So we rely on approximations.
What kind of approximations?
Well, for more accurate binding predictions, we use things like molecular mechanics, force fields, simplified mathematical models of the forces between atoms. Then we run molecular simulations, like molecular dynamics, to estimate binding free energies.
But those are slow.
They can be quite computationally intensive, yeah, especially if you want high accuracy. They're getting faster, especially with powerful GPUs, but still generally too slow for screening millions or billions of compounds. They're more useful for guiding medicinal chemistry, like predicting if a specific change to a lead compound will improve binding, or for exploring how the protein itself might flex and change shape when the drug binds.
So for screening huge virtual libraries, you need faster methods.
Right. That's where molecular docking comes in. Docking is much faster, but usually makes more simplifications. Often it treats the protein as rigid and just tries to find the best way to fit the small molecule, the ligand, into the binding pocket, ranking the different fits or poses using a simplified scoring function.
Like trying different keys in a lock.
Kind of, yeah. Figure 1.3b shows this idea of docking-based virtual screening. It's not perfectly accurate, but it's fast enough to computationally screen millions, even billions of virtual compounds. It helps prioritize which compounds are worth synthesizing and testing experimentally. It enriches your list for likely binders.
And the predicted poses themselves are useful.
Very useful. Even if the score isn't perfect, seeing how the computer thinks the molecule fits into the pocket gives medicinal chemists great ideas for how to modify it to improve interactions. You can do this interactively with graphical software, even using virtual reality now to really immerse yourself in the binding site. Figure 1 to 4, showing the HIV drug darunavir docked in the Protease active site, gives you a sense of that visualization.
And figure 1 to 5 nicely lays out that spectrum from fast, approximate docking to slower, more rigorous, free energy calculations.
Exactly. There's a trade-off between speed and accuracy, and you choose the method based on the specific question you're asking at that stage of the project.
And now artificial intelligence is being layered on top of all this.
Increasingly so, yes. AI, particularly deep neural networks, are being trained on the vast amounts of data we have. Binding data, structural data, chemical properties, outcomes from DEL screens.
And what can they do?
They're being used to predict protein small molecule binding affinity, sometimes faster or even more accurately than traditional methods for certain tasks. They can help with automated ligand design suggesting new molecules that might bind well. They're used to predict ADME properties, toxicity, even predicting protein structures themselves, like DeepMind's AlphaFold, which has been revolutionary. AI and machine learning are rapidly becoming integrated tools across the entire discovery pipeline.
Okay, so that's a deep dive into the small molecule world. But we know large molecules, biopharmaceuticals, are increasingly important.
Hugely important. Things like antisense oligonucleotides, siRNAs that silence genes, modified mRNAs like in some vaccines, and especially protein therapeutics. Monoclonal antibodies are the biggest class here, but also enzymes, peptide hormones.
Therapeutic proteins weren't really practical before recombinant DNA technology, were they?
Not easily, no. You had early examples like insulin extracted from animal pancreases, which often caused immune reactions because it wasn't human insulin, or human growth hormone extracted from pituitary glands from human cadavers, which carried the terrifying risk of transmitting prion diseases like CJD.
Yikes.
Yeah. Recombinant DNA technology completely revolutionized this in the 70s and 80s. It meant you could produce essentially unlimited quantities of highly purified human proteins or humanized versions in cell culture systems like bacteria or mammalian cells. This minimized the immune reactions and eliminated the risk of contamination from human sources.
And you can even engineer these proteins.
Exactly. It opened the door to rational design, customization, optimization. You can make human hormones like insulin or growth hormone, growth factors like erythropoietin, EPO, or GCSF, cytokines.
Right.
And then there's the massive field of monoclonal antibodies.
Used for cancer, autoimmune diseases.
All sorts of things. And these antibodies can be humanized, starting with a mouse antibody that binds the target well, and then swapping out most of it for human antibody sequences to reduce immunogenicity. Or you can even use genetically engineered mice that produce fully human antibodies directly.
These large molecule drugs generally have to be injected, right?
Yes, because being proteins or nucleic acids, they just get digested if you took them orally. And they typically target things outside of cells, like receptors or signaling molecules in the bloodstream or on the cell surface, because getting these big molecules into cells is another major challenge.
And there's optimization involved here, too.
Absolutely. You want them to be stable, avoid being rapidly degraded in the body, have low immunogenicity, and be highly potent. Strategies include optimizing how they're expressed, exploring mutations or related proteins, adding chemical modifications like pegylation to make them last longer in circulation.
Peptides are also mentioned as a growing area.
Yeah, smaller chains of amino acids. They're interesting because they can potentially tackle some of those difficult protein protein-protein interactions, those flat surfaces we talked about where small molecules struggle, computational methods are increasingly being used to help design peptides with better stability and binding properties.
Delivery is still a challenge, but evolving.
Definitely. Things like liposomal formulations are being used to protect the drug and sometimes enable different routes, maybe even non-parenteral routes like inhalation for certain biotherapeutics. It's an active area of research.
Okay, so imagine you've done all this R&D, years of work, maybe billions spent, and you finally have a promising clinical candidate, small molecule or biologic. What's the absolutely massive hurdle you have to clear before you can even think about testing it in a human being?
That would be the Investigational New Drug Application, the IND. You have to file this comprehensive package with the regulatory agency like the FDA in the US or similar bodies elsewhere. It's filed by the sponsor, usually the pharmaceutical company.
And what goes into this IND package? It sounds like a lot.
It is. It includes the whole scientific rationale for the drug, all the preclinical data, the pharmacology showing it works in models, the toxicology data from lab and animal safety studies. It needs detailed chemistry, manufacturing, and controls information, CMC, to show you can make the drug consistently and purely. And critically, it includes the detailed plan, the protocol, for the proposed first human clinical trial.
And the FDA reviews all this.
Yep. They have 30 days to review the IND and decide if the proposed trial is reasonably safe to proceed. If they don't object, you can start phase 1.
Okay, so the IND gets the green light. You move into clinical trials. What's the fundamental purpose of these trials?
The core purpose is to gather data in humans. How does the drug behave in people? It's pharmacokinetics and pharmacodynamics. And critically, to establish its efficacy, does it actually work for the intended disease and its safety profile before it can be approved for wider marketing and sale?
And the FDA's role here is obviously central in the U.S.
Absolutely. They are the federal regulatory agency responsible for protecting public health by ensuring the safety, efficacy, and security of human drugs, biologics, medical devices, and more. Their mandate also includes helping to speed innovations that make medicines more effective and safer.
And their history shows how drug regulation evolved, often in response to, well, terrible events.
Sadly, yes. The initial 1906 Pure Food and Drugs Act was pretty weak. It mainly addressed adulteration and misbranding, motivated by scandals around patent medicines, didn't require proof of safety or efficacy.
Then came the elixir sulfanilamide tragedy.
Right, in 1937, a company used a toxic solvent, diethylene glycol, to make a liquid version of a sulfa drug, killing over 100 people, many children. That led to the 1938 Food, Drug, and Cosmetic Act Amendment. This required manufacturers to submit toxicity studies and get FDA approval before marketing a new drug. A big step for safety.
But still no proof that the drug actually worked.
Correct. That didn't come until the 1962 Harris-Kefauver amendments. This was triggered by the thalidomide tragedy, where a sedative caused severe birth defects, primarily in Europe, though Dr. Francis Kelsey at the FDA famously prevented its widespread approval in the U.S. due to safety concerns.
So what did the 62 amendments mandate?
For the first time, they required manufacturers to provide substantial evidence of efficacy proof that the drug actually worked for its intended use based on adequate and well-controlled clinical trials, and also provide better documentation of the relative safety, the risk-to-benefit balance. That 1962 Act fundamentally shaped modern drug regulation.
And the FDA today faces this constant challenge, right? Balancing the need for rigorous review to protect the public against the pressure not to unduly delay potentially beneficial, even life-saving drugs from reaching patients.
It's an incredibly tough balancing act. They have finite resources and face immense scrutiny from all sides. The clinical trials themselves are conducted under strict ethical principles, like those laid out by the NIH.
Can you remind us of those principles?
Sure. Things like social and clinical value. The research has to be important. Scientific validity. It has to be well designed. Fair subject selection, not exploiting vulnerable groups. Informed consent participants must understand the risks and benefits. A favorable risk-benefit ratio. Independent review by ethics committees or IRBs. And respect for participants throughout the trial.
Okay, let's quickly walk through the standard phases of clinical trials. Phase 1.
Phase 1 is first in human. Usually a small number of participants, maybe 10 to 100. Often healthy volunteers, but sometimes patients if the drug is expected to be quite toxic, like some cancer drugs. The primary goal is safety and tolerability. You start with very low doses and carefully escalate to find the maximum tolerated dose. It's usually open label, meaning everyone knows who's getting the drug.
How long and how costly is phase 1 success rate?
Typically takes 1 to 2 years, costs maybe $10–15 million. And G&G estimates about a 50% success rate, meaning about half the drugs entering phase I make it to phase 2.
Okay.
Phase 2 is first in patient. Now you're testing the drug in a larger group, maybe 50 to 500 people who actually have the disease the drug is intended to treat. The main goals here are to get preliminary evidence of efficacy, does it seem to work, and to determine the optimal dose range.
How are phase 2 trials designed?
They are typically randomized and controlled. Participants are randomly assigned to receive either the study drug at different doses perhaps, or a control, which might be a placebo, or sometimes an existing standard treatment. They are often blinded, meaning the patients and sometimes the investigators don't know who is getting what.
And phase 2 duration, cost, success.
Phase 2 usually takes 2 to 3 years. Costs jump up, maybe $20–40 million. The success rate is lower here. Only about 30% of drugs entering phase 2 move on to phase 3. This is often where drugs fail for lack of efficacy.
Okay, so if a drug shows promise in phase 2, it moves to phase 3.
Right. Phase 3 trials are the large pivotal trials. They involve hundreds, often thousands of patients, usually studied at multiple clinics or hospitals around the world, multi-site. The main goal is to definitively confirm the drug's efficacy in a large, diverse population compared to a control group, and to gather more safety data.
Design, duration, cost, success.
They are almost always randomized and controlled, often double-blind. They are the longest phase, typically 3 to 5 years, sometimes longer, and they are by far the most expensive, costing anywhere from $50 million to $150 million, or even much more for very large trials. The success rate here is maybe 25–50%. A lot of drugs still fail in phase 3, often because the effect seen in phase 2 doesn't hold up in a larger population, or unexpected safety issues emerge.
And then there's phase 4.
Phase 4 happens after the drug is approved and on the market. It's post-marketing surveillance. It involves monitoring the drug's use in many thousands of patients in the real world. The goals are to detect rare or long-term adverse effects not seen in the earlier trials, monitor compliance, look for potential drug interactions, and sometimes explore new indications or dosing regimens. There's no fixed duration or cost for phase 4.
Looking at those phases, the length, the cost, the high failure rates, it really hammers home why developing a brand new drug, a new molecular entity, or NME, is so incredibly expensive.
Absolutely. The estimates vary, but typically range from $1 billion to maybe even $4 billion to bring one NME from the lab bench all the way through FDA approval. And that number includes the cost of all the failed projects along the way, which is most of them. The overall attrition rate from discovery to approval is huge.
So if your drug successfully navigates phase 3, showing statistically significant efficacy and acceptable safety, what's next?
Then the sponsor gathers all the data, preclinical, manufacturing, all the clinical trial results, including detailed case report forms for every patient in phase 3, and submits it to the FDA as a new drug application, NDA, for a small molecule, or a biologics license application, BLA, for a biologic like an antibody.
And this submission is massive.
Oh, enormous. Historically, truckloads of paper, now gigabytes or terabytes of electronic data. It goes through an incredibly detailed review by teams of FDA scientists, clinicians, statisticians, chemists, pharmacologists, and often involves input from external advisory committees of experts.
And how long does that review take?
It used to take years sometimes, but the Prescription Drug User Fee Act, PDUFA, passed in 1992, changed things. Under PDUFA, pharmaceutical companies pay substantial fees to the FDA when they submit an application. These fees help fund the FDA's review activities and, in theory, allow them to hire more reviewers and expedite the process. The goal now is typically a 6 to 10 month review time for standard applications, even faster for priority drugs. PDUFA also broadened the FDA's role in post-marketing safety and reviewing advertising.
If the FDA decides the benefits outweigh the risks based on all that data, they approve the drug. And then the company and FDA finalize the official label.
Right, the package insert. This is the legally binding document that summarizes all the essential prescribing information. It details the approved indications, what the drug is approved to treat, how it works, clinical pharmacology, recommended dosage, potential adverse reactions, warnings, contraindications, and sometimes includes a prominent black box warning for particularly serious risks.
But doctors can still prescribe it for uses not listed on the label.
Yes, physicians in the U.S. have the legal right to prescribe approved drugs off-label for uses, doses, or patient populations not specifically included in the FDA-approved label if they judge it's medically appropriate based on scientific evidence or clinical experience. However, insurance reimbursement for off-label uses can be tricky, and there can be increased liability risks for the physician. Companies are generally prohibited from promoting off-label uses.
A really critical question hangs over all this. How exactly does the FDA determine if a drug is safe and effective? What's the standard?
Well, safe is always relative, right? No drug is perfectly safe. It's about whether the demonstrated benefits outweigh the known and potential risks for the specific approved indication. For effective, the standard since 1962 is substantial evidence derived from adequate and well-controlled investigations.
And what does adequate and well-controlled usually mean in practice?
Typically, it means the FDA wants to see results from at least 2 independent, large, Phase 3 randomized double-blind controlled clinical trials that show a statistically significant and clinically meaningful benefit compared to the control group. The control is often a placebo, but can also be an active comparator, an existing standard treatment.
There's an ongoing ethical debate about using placebos when effective treatment already exists, isn't there?
Yes, definitely. The Declaration of Helsinki, an international ethical guideline, raises concerns about giving patients a placebo if a proven effective therapy is available. Proponents of placebo controls argue they provide the clearest, most unambiguous measure of a new drug's absolute effect, especially if the existing treatments have only modest benefits or significant side effects. It's a complex issue with different viewpoints.
And another layer of complexity is what you actually measure in these trials. Surrogate endpoints versus true clinical endpoints.
Right. A true clinical endpoint is a direct measure of how a patient feels, functions, or survives. Things like preventing heart attacks, strokes, bone fractures, reducing tumor size, or improving survival time. These are the outcomes that matter most to patients.
But they can take a long time to measure.
Exactly. So often, trials rely on surrogate endpoints instead. These are laboratory measures or physical signs that are thought to correlate with or predict a true clinical endpoint, but are usually easier and faster to measure.
Like LDL cholesterol levels for heart disease risk.
That's the classic example. Or blood pressure. Or bone mineral density for fracture risk. Or HbA1c levels for diabetes complications. Or viral load for HIV progression. Using surrogates can dramatically shorten trial duration and reduce cost.
But the risk is they might not actually predict the true outcome accurately.
That's the big caveat. As G&G points out, sometimes a drug can improve a surrogate endpoint but fail to deliver the expected benefit on the true clinical endpoint or even cause harm. The elamide story is a perfect illustration.
Remind us.
Ezetimibe, Zetia, was approved based on strongly lowering LDL cholesterol, the surrogate. But then an early trial failed to show it improved carotid artery thickness, another surrogate, casting doubt. It took a massive multi-year trial called Improve It to finally show that adding ezetimibe to a statin did reduce the rate of heart attacks and strokes, the true endpoints, compared to the statin alone. It vindicated the drug, but after years of uncertainty.
And the torcetrapib example shows the potential downside even more starkly.
Yeah, torcetrapib raised HDL, good cholesterol, another surrogate, but unexpectedly increased mortality in clinical trials. A disastrous outcome highlighting that biology is complex and relying solely on surrogates can be risky. The FDA often requires post-approval studies to confirm clinical benefit when initial approval is based on a surrogate.
Drug safety seems like perhaps the biggest ongoing challenge, especially after a drug gets approved.
It really is, because, again, no drug is perfectly safe for everyone. They all have potential side effects. The major challenge is detecting the rare adverse effects. Things that might only happen in, say, 1 in 5,000 or 1 in 10,000 patients.
And Phase 3 trials usually aren't big enough to reliably detect those.
Exactly. Phase 3 might enroll a few thousand patients. That's generally not large enough or long enough to pick up very rare, but potentially serious side effects.
So the true full spectrum and incidence of rare side effects often only become apparent after approval once millions of people start using the drug in the real world.
That's right. Detecting those rare effects pre-approval would require impossibly large and long clinical trials, which would add enormous cost and massively delay access to potentially beneficial therapies. So there's an inherent societal trade-off between accepting some level of risk for rare events versus the cost and delay of trying to find every last one beforehand.
And product liability lawsuits are a significant factor in this landscape.
Huge factor. Companies can be sued if their product is deemed defective due to design flaws, manufacturing problems, deceptive marketing or promotion, violating regulations, or, critically, failure to adequately warn about risks.
Even if the FDA approved the drug in its label.
Yes. Courts have found companies liable for failure to warn, even if the FDA had approved the warning label, if new information emerges or if the warning was deemed insufficient. And specifically, courts have held companies responsible for the warnings included in direct-to-consumer advertising. The Wyeth v. Levine Supreme Court case in 2009 was pivotal here.
What did that case establish?
It affirmed that FDA approval of a label does not necessarily shield a manufacturer from state-level lawsuits alleging inadequate warnings. Basically, states could potentially impose stricter warning requirements than the federal government. This creates ongoing friction and uncertainty for manufacturers.
And this fear of liability must have consequences.
It likely does. It might make companies overly cautious in testing, leading to delays, increased development costs, more trials and data needed for legal defense, and potentially act as a disincentive for developing drugs for rare diseases or small populations where the market is small but the liability risk remains. Liability for rare, undetected adverse effects is a constant point of tension, often resolved case by case through litigation or settlements.
So how are these rare adverse events detected after marketing? What are the systems?
There are formal pharmacoepidemiologic methods like large cohort studies comparing users and non-users, case control studies looking back at exposures among people who had an event, and meta-analyses pooling data from multiple studies. But equally, or maybe even more important, are the voluntary reporting systems.
Like MedWatch in the U.S.
Exactly. The FDA's MedWatch program allows physicians, pharmacists, nurses, consumers, basically anyone, to voluntarily report suspected adverse drug reactions. Companies are also legally obligated to report any adverse events they become aware of to the FDA. These spontaneous reports, even if imperfect, can act as crucial early warning signals for potential safety problems that weren't apparent in clinical trials. Hospital safety committees and third-party payers also contribute data.
Okay, moving from population-level effects to individual variation. Personalized medicine or precision medicine is a huge theme now.
Yeah, individualized medicine, precision medicine. The core idea is to move away from a one-size-fits-all approach and better tailor treatments to the individual patient's unique characteristics, recognizing that people vary tremendously.
In what ways?
In how they respond to drugs, efficacy, their susceptibility to side effects, how they absorb, distribute, metabolize, and excrete drugs, ADME, the underlying biological cause, etiology of their disease, their environment, and crucially, their genetics.
And advances in genomics have really fueled this.
Absolutely. The cost of sequencing a human genome has plummeted, down to maybe around $1,000 now. This allows researchers to analyze vast amounts of genomic data alongside clinical data, phenotype, to understand the genetic basis for variability in drug response and toxicity.
Biomarkers are key tools here.
Critical tools. Biomarkers are measurable indicators, could be a gene variant, a protein level in the blood, an imaging result that can help guide treatment decisions. They might be used to monitor treatment response, predict who is likely to succeed or fail on a particular drug, anticipate who is at high risk for a specific side effect, or help select the optimal dose or drug choice for an individual.
This seems especially powerful in cancer treatment.
Definitely. Targeting drugs specifically to the genetic mutations driving a patient's tumor is a cornerstone of modern oncology. For example, testing for HER2 mutations to guide use of Herceptin in breast cancer, or EGFR mutations for certain lung cancer drugs. Using biomarkers to select patients for clinical trials can also make trials more efficient and help define more clearly the population that will actually benefit from the drug if it's approved. G&G dives deeper into this in later chapters on pharmacogenetics and post-marketing drug safety.
Okay, so all this incredible science, technology, clinical testing, and regulation doesn't happen in a vacuum. It operates within a larger context of public policy, economics, and societal values.
And this is where you see some fundamental tensions play out. On one hand, many people view access to necessary medicines as a basic right, part of healthcare, and are critical of the profit motive in this space. On the other hand, in a capitalist system like the U.S., drug development is largely carried out by private, investor-owned companies whose primary driver is profit, and who take on enormous financial risks to develop these products.
Critics often have a pretty negative view of the pharmaceutical industry.
Yeah, it's often portrayed as greedy, prioritizing profits over patients, focusing on Me Too drugs instead of true breakthroughs. Yet the current system, at least in the U.S., largely relies on this profit incentive to fuel the incredibly expensive and risky process of innovation. Without it, who would invest the billions needed? It's a scientific process, but also deeply political and economic.
And the high cost of drugs is probably the biggest flashpoint.
Absolutely. It's a huge concern for consumers paying out of pocket, for private insurers, and for government programs like Medicare and Medicaid, especially for some new specialty drugs like certain cancer therapies or cures for diseases like hepatitis C, where the launch prices can seem astronomical, potentially exceeding the direct cost of manufacturing by orders of magnitude.
And the U.S. situation is unique.
It is among large developed countries in that the federal government generally doesn't negotiate or control drug prices directly. Price is not formally considered as part of the FDA approval process itself. This often leads to U.S. consumers paying significantly higher prices for the same drugs compared to people in other countries where governments do negotiate prices.
The argument being that U.S. prices effectively subsidize R&D costs for the rest of the world.
That's often the argument made, yes. And it's a major point of political debate. The hepatitis C example highlighted in Box 1.3 is a perfect case study.
What happened there?
A new generation of drugs came out that offered a true cure for hepatitis C, which was amazing, but they launched with extremely high price tags, like $80,000 to $90,000 for a course of treatment. This led to huge controversy, insurers initially refusing to cover it for many patients, fierce debate about cost versus value, rationing of care, and eventually prices did come down somewhat as competing drugs entered the market, allowing for negotiation. But it really crystallized the difficult questions around affordability, access, and how we value therapeutic breakthroughs.
These are really deep pharmacoeconomic questions.
They are. How do you compare the cost of a drug to the potential downstream savings from avoiding hospitalizations, liver transplants, or lost productivity? How do you put a dollar value on improved quality of life or the convenience of an easier dosing regimen? And the toughest one, what constitutes a fair profit margin for the companies given the risks they take? There are no easy objective answers. Market forces, negotiation, and government policy all play a role.
The industry, of course, counters that high prices are necessary to recoup those massive R&D investments and fund future innovation.
Right. They emphasize the $1 billion cost per approved drug, the extremely high failure rate, the fact that most revenue comes from only a few blockbuster drugs that have to pay for all the misses. They also point out that while prescription drugs are a noticeable part of overall healthcare spending, maybe 9–13% in the U.S., according to G&G, the growth in drug prices overall has often been slower than growth in other parts of the healthcare system, like hospital costs. So even drastic cuts to drug prices might not solve the larger healthcare affordability crisis.
They also stress the inherent riskiness of the business.
Definitely. High upfront R&D costs, very low probability of success for any given project, a relatively short period of market exclusivity due to patent life and development time, stringent regulation, the constant threat of product liability lawsuits, and intense competition from other companies. From a business perspective, the U.S. free market system tends to reward high-risk ventures with the potential for high rewards, and pharma certainly fits that description. But balancing that with affordability and access remains a central challenge. There's growing pressure, politically and socially, to link prices more explicitly to the therapeutic value a drug provides and the unmet medical need it addresses. But assessing that value is complex.
Who actually pays for these drugs in the U.S. system?
It's a mix. Consumers pay some out of pocket through copays and deductibles. Private health insurance plans cover a large portion, and public programs, primarily Medicare for seniors and Medicaid for low-income individuals, plus programs like SEIP for children, cover a very significant chunk, meaning taxpayer dollars are heavily involved. Over a third of retail prescription costs are paid by public funds. Generic drugs play a crucial role in containing costs once brand name drugs lose patent protection.
This links directly to intellectual property and patents, which seem foundational to the whole R&D model.
Absolutely fundamental. IP protection, mainly through patents, is seen as the vital incentive for companies to invest billions in risky R&D. Abraham Lincoln is quoted, "Patents add the fuel of interest to the fire of genius." A U.S. patent typically grants the inventor exclusive rights to make, use, and sell the invention for 20 years from the date the application was filed.
But the effective market exclusivity is much shorter.
Right, because it takes many years, often 10 or more from patent filing, to get through development and FDA approval. So the actual period of market exclusivity before generic competition can start might only be around 10–12 years on average. The Hatch-Waxman Act of 1984 allows for some patent term extension to partially compensate for delays during FDA review, but it's still a limited window.
Once the patent expires, generic competition enters.
Yes, and generic manufacturers can get approval through an abbreviated pathway. They don't need to repeat the large clinical trials. They mainly need to demonstrate therapeutic equivalence, that their product contains the same active ingredient in the same amount and dosage form and produces essentially the same concentrations in the bloodstream, bioequivalence, as the original brand name drug. Because their R&D costs are minimal, generics can be sold much, much cheaper, driving prices down dramatically.
This leads to ongoing debates about optimal patent length, doesn't it? Should patents be shorter to lower costs sooner, even if it risks stifling innovation or forcing higher launch prices? Or should they be longer to give companies more time to recoup costs, potentially allowing lower prices over time?
Exactly. There are arguments on both sides. And of course, a patent only provides protection if your drug remains competitive. If a clearly superior drug comes along from a competitor, your patent might not be worth much anymore.
What was the impact of the Bayh-Dole Act of 1980?
That was a significant piece of legislation. Before Bayh-Dole, the IP rights for inventions made using federal research grants generally belonged to the government. Bayh-Dole transferred those rights to the universities and academic scientists who actually did the research. The goal was to incentivize them to patent their discoveries and partner with industry to translate basic research into actual products for the public benefit.
Did it work?
It's widely credited with boosting technology transfer from universities to industry and spurring innovation, leading to many new drugs and technologies. However, it also raised concerns about potential conflicts of interest, blurring the lines between academic research focused on knowledge generation and commercial interests focused on product development.
Now, for biologics, the large molecule drugs, the concept of a generic is more complicated.
Much more complicated. Small molecules are relatively simple chemicals. You can make identical copies fairly easily. But biologics proteins, antibodies, are large, complex molecules produced by living organisms, cells. Their exact structure, including subtle variations like folding and post-translational modifications, like adding sugars, can significantly impact their function, pharmacokinetics, and importantly, their potential to trigger an immune response, immunogenicity.
So proving two biologics are truly identical is much harder.
Extremely difficult, maybe impossible in some cases. That's why we don't have generic biologics in the same way we have generic small molecules. Instead, we have biosimilars,
which were enabled by a specific pathway.
Yes, the Biologics Price Competition and Innovation Act, BPCIA, which was part of the Affordable Care Act, ACA, in 2010. It created an abbreviated licensure pathway for biological products demonstrated to be biosimilar to an already FDA-approved biologic, the reference product.
What does biosimilar mean exactly?
The standard is that the product must be highly similar to the reference product, notwithstanding minor differences in clinically inactive components, and there must be no clinically meaningful differences between the two in terms of safety, purity, and potency. Proving this typically requires extensive analytical data comparing the structures, animal studies, and usually some clinical data in humans, although generally less than required for the original biologic. The interpretation of highly similar and no clinically meaningful differences is still evolving and subject to debate. Biosimilars offer potential cost savings, but the development and approval process is much more complex and expensive than for small molecule generics.
Okay, shifting gears a bit. Beyond R&D and regulation, how drugs are promoted and marketed is another major policy area.
Yeah, and it's often controversial. Ideally, physicians would get all their information from objective sources like peer-reviewed journals and unbiased continuing education. But in reality, pharmaceutical companies spend enormous amounts of money promoting their products.
How do they do that?
Through advertisements in medical journals, sponsoring educational events, sending pharmaceutical sales representatives, pharma reps, to visit doctors' offices, providing free samples, and most visibly to the public through direct-to-consumer DTC, advertising on TV, radio, print, and online.
And the spending on promotion is huge.
Often reported to be comparable to or even exceed spending on R&D, although the accounting can be debated. The industry faces significant criticism for its marketing practices. All promotion is supposed to be consistent with the FDA-approved label and provide a fair balance between claims of benefit and information about risks and side effects.
DTC advertising seems particularly contentious.
It is. The US and New Zealand are the only developed countries that allow broad DTC advertising of prescription drugs. Canada allows a limited form. Critics argue that DTC ads can medicalize normal life experiences, overemphasize benefits while downplaying risks, drive demand for expensive new drugs when cheaper, older options might be just as good, and put pressure on doctors to prescribe specific drugs requested by patients.
What's the counterargument?
Proponents argue that DTC ads educate patients about diseases and potential treatments they might not have been aware of, reduce stigma around certain conditions like depression or erectile dysfunction, and empower patients to have more informed conversations with their doctors, potentially leading to earlier diagnosis and treatment. Research shows DTC ads do influence patient requests and physician prescribing.
And what about influencing physicians directly? Sales rep.
That's another area of concern. There used to be widespread practices of giving gifts, funding lavish trips, etc. Those have been curtailed significantly by industry codes, like the PHRMA code updated in 2009, and institutional policies. Overt gifts are now largely banned, but things like company-sponsored meals, often linked to educational presentations, though sometimes the educational component is minimal, and payments to physicians for consulting or speaking engagements are still common and raise concerns about potential bias in prescribing. The number of sales reps has decreased somewhat amid this scrutiny.
Finally, the chapter touches on some difficult issues of global justice and ethics.
Yeah, this is important. Because drug development is so expensive and largely driven by market potential, research inevitably tends to focus on diseases that are prevalent in wealthy countries, where companies can expect to recoup their investment and make a profit.
Meaning diseases primarily affecting poor countries get less attention.
Often, yes. Drugs for neglected tropical diseases, or even common diseases that primarily affect populations in low-income countries, may not offer sufficient commercial incentive for large pharmaceutical companies. Development for these often relies heavily on public funding, like NIH grants, or philanthropic organizations like the Gates Foundation.
And patent laws can also create barriers to access in developing countries.
That's a major issue. Patents granted in the US or Europe can make new, life-saving drugs unaffordable in countries where the vast majority of people cannot pay those prices. There are mechanisms like compulsory licensing that countries can sometimes use, but it's politically contentious.
Adding another ethical layer is where clinical trials are conducted.
Right. Companies are increasingly conducting clinical trials, especially later phase trials, in developing countries. G&G notes a roughly 2,000% increase in foreign clinical trials for U.S. drugs over a 25-year period.
Why is that?
Reasons can include lower costs, faster patient recruitment, sometimes because access to healthcare is otherwise limited, and potentially less stringent regulatory oversight in some places, although ethical standards are supposed to be maintained.
But the ethical concern is?
The concern, highlighted by principles like those in the Belmont Report, is about justice. Are the populations participating in the research likely to benefit from the results? If a trial proves a drug is effective in a low-income country, but the resulting approved drug is then priced out of reach for most people in that country, have the trial participants been exploited? It raises difficult questions about who bears the risks of research versus who reaps the benefits.
What's the counterargument there?
The counterargument often made is that participating in clinical trials may be the only way for people in some resource-poor settings to access any kind of advanced medical care, diagnostics, and monitoring, even if it's within the context of a trial. So the trial itself provides a benefit. It remains a very complex and controversial issue.
This kind of brings us full circle, back to the debate about Me Too drugs versus true innovation and the overall productivity of the R&D enterprise.
Exactly. We talked about Me Too drugs, structurally similar derivatives of existing drugs, sometimes called follow-on drugs. They might be developed by competitors trying to get a piece of an established market, or sometimes even by the same company trying to extend market exclusivity, like the esomeprazole omeprazole example in Box 1-4.
And the criticism is they don't add much value.
Critics argue they often offer only marginal, if any, benefits over existing therapies, but are expensive, contribute to rising healthcare costs, and divert R&D resources away from riskier projects aimed at true breakthroughs for unmet needs.
But as you said earlier, they can offer real value sometimes.
They certainly can. For some patients, a follow-on drug might genuinely work better, have fewer side effects, or be easier to take, leading to better compliance. Sometimes a Me Too drug turns out to have advantages that make it best in class, like atorvastatin did in the statin market. So it's not always black and white.
G&G presents some data on the actual output, the number of new drug approvals over time. What does that show?
Figure 1 to 7 shows that for a long period, from roughly 1980 to 2016, the number of approvals for new molecular entities, NMEs, was relatively modest and fairly flat, averaging around 24 per year in the U.S. There has been a noticeable uptick more recently in the 2017-2021 period, with an average of about 38 NMEs and 13 new biologics approved per year, so some increase.
But how does that compare to the investment in R&D over the same period?
That's the striking contrast. Over that same time frame, roughly 1980 to 2021, industry R&D spending absolutely exploded G&G cites a 45-fold increase from around $2 billion per year to over $90 billion per year.
Wow, so a 45-fold increase in R&D spending, but maybe only like a doubling at best in the rate of new drug approvals recently. There seems to be a disconnect.
There does. Despite all the incredible technological advances we've discussed, combinatorial chemistry, the human genome sequence, HTS, structural biology, computational methods, molecular biology, genetics, the overall productivity of the R&D enterprise, measured purely by the number of approvals per dollar spent, seems to have declined significantly over recent decades. It suggests the process remains incredibly difficult, complex, and inefficient, or perhaps that the low-hanging fruit targets have been picked. Or maybe these newest technologies are still too new to have fully translated into a massive surge in approvals yet.
And companies face huge pressure to improve this productivity, especially as patents on their existing blockbuster drugs expire.
Absolutely. They need to refill their pipelines. The argument G&G makes is that the path forward likely involves moving towards more targeted, individualized therapies.
Using diagnostics to identify the right patients.
Exactly. Using molecular diagnostics to understand the specific basis of a patient's disease and select treatments most likely to work for them. This could improve clinical trial success rates by enrolling only likely responders, lead to better patient outcomes, and ultimately might be crucial for the survival of companies in an increasingly challenging environment.
The hope, then, is that these modern techniques, even if they haven't caused a huge surge in approvals yet, will eventually lead to more effective, more specific treatments.
That's the hope, that the deeper understanding of biology combined with these powerful new tools for chemistry, computation, AI, genetics, and molecular medicine will ultimately translate into therapies that are truly transformative for patients. But it's clearly a field still in major flux, where cutting-edge scientific possibility is constantly bumping up against hard economic realities and complex societal expectations.
So what does all this mean? Wow. We've just navigated a truly deep dive, guided by GOODMAN and GILMAN, covering this incredibly complex journey of a drug. From those ancient observations of plants, all the way to today's AI-driven design labs, then through that intense gauntlet of preclinical testing and multi-phase clinical trials overseen by regulatory bodies like the FDA.
Right.
And finally landing in this intricate policy world of pricing, patents, promotion, ethics, and global access.
It's just so much more than finding a useful molecule, isn't it? It's this multi-decade, multi-billion-dollar undertaking. It's fraught with scientific uncertainty, immense technical challenges, huge regulatory hurdles, and these really deep ethical and economic questions at every stage. To really get your head around pharmacology, you have to understand this entire ecosystem that produces the drugs in the first place.
Which really brings us to a final important question to leave you with. As our scientific capabilities continue to accelerate, AI designing molecules, genetic insights revealing individual patient differences, entirely new modalities like gene and cell therapies emerging. How are we, as a society, going to grapple with those fundamental tensions this chapter lays bare? How do we ensure that these increasingly powerful, complex, and often incredibly expensive innovations get developed efficiently, sure, but also, crucially, how do we make sure they're accessible and affordable for everyone who needs them all around the world? Balancing that need for innovation with the demands of equity and affordability, that's a huge challenge going forward. Definitely something to think about.